So what I'd like to do is first go over a little bit of background on autism and why autistic children may be at higher risk for environmental insults, and then progress to the issue of mercury, which has been almost the limelight of autism and environmental risk, and then move to some of the newer risks that we feel are probably understudied. So this is by way of review. I think most of you have a good grasp of how autism is diagnosed. It's a severe pervasive impairment. It has a very defined definition by the American Psychiatric Association in their DSM-4 revised version, which is about to become the DSM-5. But basically, the way you diagnose autism is based on behavioral outcomes and reaching behavioral landmarks. So it's a severe and pervasive developmental disorder, but the core trigger for getting an autism diagnosis is impairment in reciprocal social interactions. There are also associated impairments either in verbal and nonverbal communication skills, the presence of stereotypic behavior and activities, and relevant to environmental exposures is the fact that children with autism tend to be much more in contact with their environment. They tend to have much more hand-to-mouth interactions. They tend to be much more intimate with the floors, the couches, and if they're outside with their natural environment. There are early onset forms of autism, and if you were next door, you'd kind of get the latest state-of-the-art lecture on that. In some cases, when we think about regression, if you looked carefully enough earlier on, there may have been some indicators. But there are some individuals that really show no signs of impairments. They hit the trajectory, the trajectoral hallmarks, but sometime around 18 to 36 months, begin to lose skills that they've gained. This is called regression. And we think of this particularly in terms of postnatal exposure to chemicals that might either trigger regression or contribute to regression, the severity of regression. Now, in addition to the core symptoms that define autism, it's really important to note that there are many comorbidities. And here, from a toxicological perspective, we know very little about chemicals that change social behavior, mainly because if you do an epidemiological study, the population sets that have been studied up until very recently aren't large enough to make any really major conclusions. There's just not enough statistical power. Again, not that autism is rare, it's 1 in 110, but from an epidemiological perspective, if you want to study gene-environment interaction, you need very, very large populations to study, and those just haven't been done. And most studies in epidemiology focus on cognitive endpoints rather than social endpoints. Now, if you flip that around and you say, well, we have animal models and we have cell models, but why are those somewhat deficient when you're thinking about major social deficit as an outcome, major language deficits as an outcome? I've tried many interbreeding strategies amongst the mice in my lab, but none of them have developed language. So they seem to fall a little short. They are social, but their sociability is mainly through scent. And so, you know, there is quite a bit of controversy as to how good model mice can be for social interaction endpoints. But having said that, there really haven't been a huge number of studies that have looked at how chemicals influence social outcomes even in these animal models. However, if you think about all of these comorbidities that are really quite frequent in autism, such as cognitive deficits, anxiety disorder, seizure disorders, sleep disorders, and GI disorders, we do have data from animal models, from cellular models, that look at these endpoints. And so we look at these maybe not so much as comorbidities as a clinician might view it, but we look at it more as giving us an indication of which chemicals may in fact be triggers or where susceptible individuals may have a much higher sensitivity to these kinds of outcomes. The other tip we should get in studying chemicals is the fact that autism has a 4-to-1, approximately 4-to-1 distribution. So there's a gender bias in the prevalence of autism. Males are more frequently diagnosed with autism than females. How many of you have heard about endocrine disruption and the problem that we now face with endocrine mimics? It's a big deal in environmental health sciences these days. Many of the chemicals, and I'll show you some, that have been put out and considered to be safe have been shown, once use has brought their concentrations up in the environment and brought their concentrations to detectable levels in human tissues, have been shown to be endocrine disruptors. They disrupt the thyroid system, the estrogen system, and the other hormonal systems in humans and in animals. So I think that we have a lot of leads. The question is, how do you study this? When we set out about 11 years ago, just about the time the Mind Institute started to come together, the idea in the field, which was heavily dominated by behavioral sciences, was that autism, in fact, was one of the most heritable disorders known, complex developmental disorders known. And in many cases, the citations went something to the effect, well, you know, there's no room for environment in autism because if you look at monozygotic twins, identical twins, the concordance rate for an autism diagnosis is greater than 80%. And in many cases, the stated values were actually greater than 90%. This was about 10, 11 years ago. Okay? The reciprocal argument was that the concordance rate for dizygotic twins, non-identical twins, was, low, 10%, 12%. And that made a very convincing argument, but you have to remember that once something becomes dogma in science and nobody challenges it with a new study, it pretty much is enduring. Well, this sort of changed on July 4th of 2011 with this paper. This is Otto Kaiser, and there was a couple of people from the Mind Institute involved on this paper, but it sort of made a very important point. If you have a very well-studied group of individuals, like these individuals that were studied out of the Kaiser database, you find something that actually goes against the dogma, that concordance rates aren't 90%, that concordance rates are closer to 60% for monozygotic twins, somewhere between 50 and 60%. That's quite a bit lower than the dogma. Moreover, if you looked at dizygotic twins, whether they were male or female, the concordance rate was more than 10%. In other words, they were up around 30%. So, monozygotic concordance lower than what we had predicted, and dizygotic concordance higher than what we had predicted. What's one conclusion that you can come from that? Well, maybe heritability, genetics, is only playing a part, and shared environment plays at least as important a part in determining risk. So, the conclusion from this paper, which immediately caused a reaction in the scientific world, was that in fact shared environment was at least as important as genetic heritability in dictating autism risk. So now we got new values here. There's no question that autism risk is very complex. It involves many genes. It's probably several disorders that converge on a common behavioral diagnosis. But we need to now start paying more attention, not so much on the genes, and I'll say a little bit more about that, but on the environment. And we're not just talking about the bad things that power plant is spewing out, but we're talking about the whole cadre of how we've changed our lives, from where we live, how we live, what we eat, what we supplement with, and of course, the kinds of chemicals that are getting into our bloodstream. So in terms of genetics, there are a few highly penetrant known mutations in humans that confer a very high risk to autism. And I've listed a few here. Fragile X has a very high autism rate, but it should be noted that there are some children with Fragile X that do not have autism. What makes the difference since it's the same gene? That's a tip. It's trying to tell us something. There are also some very rare mutations. They're almost called personal mutations, which are so penetrant that essentially they do cause autism. And a classic example, it was only discovered in 2006, but it's now classic in the sense that it's one mutation. It's called Timothy syndrome. It's a mutation in a voltage-dependent calcium channel. And the researchers in Boston would have never picked this up, except the kids get what is called long QT syndrome, which causes heart arrhythmias. Somebody very insightful in a clinic realized that as these kids came in, because there was a study on Timothy syndrome in Boston at the time, that a lot of these kids, like 60% of them, were autistic, met the diagnosis for autism. And so you scratch your head and you say, what's a calcium channel in the heart doing, promoting autism risk? Especially since it's not a missing gene, it's a gene with a single missense mutation, one amino acid substituted for another. But it's causing that channel, that ion channel, to misbehave. And the real diagnostic presentation is long QT. Only after they realized that these kids had a 60% autism rate did they suggest it was in fact a risk factor for autism. From the biology, you say, well, you know, it may not be such a strange and unexplainable phenomenon. That same channel is expressed in critical regions of the brain and is involved in brain development. It's also expressed in immune cells. And so if you go back to this idea that autism has immunological contributions, sleep disorder, seizure disorder, you can start to say, well, here's an example, Timothy syndrome, where essentially it's a who's who of these symptoms, because children with Timothy syndrome have huge immune problems, they have autism, they have seizure disorders, okay? But again, this is a very rare disorder, one in, I think, somewhere between 10 and 30,000, okay? And then there are new ones that have come up, like the neuroligin mutations. Those are even rarer. Those are really almost kindred specific, that one family has passed this on within their kindred. Most autism is idiopathic. What does that mean? It means we actually don't know what's causing the autism. And so there are general ideas of how this comes about. So the leading sort of theories on idiopathic autism is that something causes copy number variations or changes in copy number burdens within chromosomes of the DNA. We just don't know how that works. There's an error in replicating the DNA in such a way that you either get deletions or duplications, and that causes problems. These could be inherited. There are examples of this from mom or dad. But now we also know that these could be de novo, which means that they don't appear in either biological parent. They're actually appearing in the child for the first time. There's a lot of interest in how that happens, because that can, in fact, be environmentally driven. Another way that you can get environmental changes in our genome is through epigenetic changes. So you're probably quite aware that over the last 30 to 40 years, the prevalence of autism has gone from about 1 in 10,000 to the current 1 in 110. Genetic mutations, the changes in the human genome, cannot possibly occur at such a high rate. So there must be other ways to change outcome in the human genome. And this is the scary part. At least it scares me. Because how you change the methylation patterns, you chemically modify the DNA through small changes like methylation or acetylation, and which regions of the DNA get methylated or acetylated, or maybe don't get methylated or acetylated, can go back and affect copy number variations. And once these changes are instated, they're permanent because they can be transmitted from generation to generation. So there's a lot of attention now being focused on epigenetics and how epigenetics may in fact influence things like copy number burden and copy number variations. So there's a lot of room for gene-environmental interactions here. So what do we know about the genetics based on standard protocols? So we have invested a huge amount in the genetic approach to understand autism, as well as many other neurodevelopmental disorders. Are there certain sets of genes that we know are going to be very reproducible biomarkers of autism risk? Well, if it's Timothy syndrome, yes. Neuroligin, there's some question. Fragile X, yes, but it's only a fraction of the kids that develop autism, so there's something else going on. So we get to this. So if you put together, as Dan Geschwin and Abrahams did in this review, 18 large studies on association, genomic-wide associations, SNP analysis that use SNP arrays, which just query a very large number of genes for mutations or polymorphisms, or you even use these other types of arrays that look for copy number variants. They're called CGH arrays. What you find is that every chromosome has a huge number of possible risk loci. So the bottom line is that autism's genetically is very complex. So I sat there and looked at this and I said, well, I'm a little confused. If we're going to think about 80,000 industrial chemicals and a very large number of genetic possibilities, I don't think I'm smart enough to figure it out. Okay. But there's very recent information coming out now from these approaches. The CNV arrays, the CGH arrays, the SNP arrays, they have a problem. They don't match up when you look across platforms. And these were made on, there are several companies that provide these arrays. And depending on who you compare to who, the concordance rate on the array readout can be as poor as 50%. So you have a 1 out of 2 chance that whatever you found was just by chance. That's not a good hit rate, okay? So this may be a little misleading in the sense that if you replicate, maybe you don't find this particular location, okay? And that's something that people are working on very hard now to try to improve this, to try to equalize this and to say what's the noise and what's real, okay? One way to get around this is very, very recent technology. There are now facilities being put in place, and one of them is going to be UC Davis, where you can now not use the array approaches, but essentially do very deep sequencing of the entire genome, so you have nucleotide level information. And the error rates with those are much lower. The false discovery rates, so positives that are not positive and negatives that are not negative, very, very low, less than 10%. Okay? So that improves the quality of the data. So that's as much as I want to say about genetics until a little later when I talk about specific chemicals. But here, essentially, the genetics is still in flux, and we may want to question much of the results that have already appeared in the literature based on errors. Environment. There was only one environmental trigger when we started working on autism 11 years ago, at least of concern to parents and advocacy groups. It was mercury, and in particular, mercury in vaccines. Why was mercury in vaccines? Because in the manufacturing process, there's two ways you can prepare the vaccine. One is a single-use vial, which essentially is sterile. You use it for one patient, and then you toss the vial away. The other formulation was called a multi-use vial, where you get quite a bit of vaccine in there, and you don't want to throw the unused portion away, so you save it for the next patient. And so there are typically 5 or 6 doses in any given vial, and because you're going in and out of that vial, even though you take great care not to introduce bugs in there, you needed a preservative, especially for countries where refrigeration wasn't quite a standard operating procedure. And so everybody, whether you had the multi-use vaccine in the United States or they were shipped abroad, essentially they were the same vaccine, got about 50 micrograms of mercury in a particular type of vaccine. For example, the DAP. Okay. Now, the MMR, which makes a lot of news about vaccines and autism, was never in a multi-use formulation and so never contained mercury. And so even back then, there was some confusion about, well, is it the MMR vaccine or is it the mercury in the MMR vaccine? What exactly are we talking about, okay? And so as you can imagine, there's a lot of public press and concern about mercury in vaccines, but then also about vaccination per se. And so that really dominated the environmental side of autism risk. But we now know that when mercury was taken out of the vaccines around 2000, slowly weaned, the autism rates continued to go up, and I'll show you that data in a second. So to a neurotoxicologist, which I consider myself, putting mercury in an injectable form, somebody wasn't thinking. Why? Because we know that mercury, there are many examples showing, unfortunately from tragic mistakes, that mercury is a very, very potent developmental neurotoxicant. But there's something else going on, because when you've taken what you think is a major contributor to the increasing prevalence of a disorder, and the disorder continues to rise, it's an indication that you should be looking in other places as well, okay? So here are some examples. We've pushed to older and older ages when people have children, and so now there are several sound epidemiological study that suggests that maternal and paternal age do play a role in autism risk. Can it account for the rise to 1 in 110? No. They're more subtle contributions. Proximity to sources of pesticides, and I'm going to spend some time on this one. Proximity to major highways. That paper was published last year and actually did get quite a bit of press attention. And I'll say that when we look at these two as possible risk factors, to a toxicologist you say, but of course, because the compounds in pesticides do affect the same systems that are impaired in autism. And I'll give you a specific example. What's the major pollutant near major highways? It's combustion products. We know that these contain very high levels of polyaromatic hydrocarbons. Well, one of the hottest genes to be associated with autism risk, the MET gene, actually is regulated by a promoter, thing that regulates its transcription, which is extremely sensitive to polyaromatic hydrocarbons. So you can start to piece together the science. And I'll say more about that in a second, okay? And then very recently, Something that isn't exogenous, but something that's a nutrient, is folic acid supplementation. That the group at Davis basically found that there was kind of a very protective effect if women supplemented with folate within a certain period of time before conception. So let's talk a little bit about the chemicals. So let's say that you have a pesticide. Let's call it an insecticide, because Rondi talked about picking the aphids off of, because you can't apply the spray stuff. How many of you think that because it's a poison, obviously engineered to kill something like a flea or a tick or spider, and they're really good at it because you know if you spray them, if it's a pyrethroid, that bee or that hornet is going to just fall right out of the air immediately. That's the way the insecticide works. And then it's going to do this business, this kind of dance, and eventually it'll either desiccate or die from a neural problem. How many of those have gone thorough developmental neurotoxicity testing in the United States? Let's show of hands. Greater than 90%. Oh, you guys know way too much. Greater than 50%. 10%? Yeah, so I think you're on board. Very few. Only the ones that are now either entering the market for the first time for registration, or ones that have come up, used up their lifetime and are being re-registered, are being required by the EPA to undergo developmental neurotoxicity testing. Usually the test for neurotoxicity is you give them a huge dose. If the mice don't immediately go into seizures and die, sounds good. They might get to market. And we now know that that's not the right endpoint. We know that these chemicals can have much more subtle effects that could contribute to cognitive delays, language delays, developmental disorders. So we've come up against a real problem because we have virtually 80,000 important industrial chemicals, and there is no way we're going to test all of these for developmental neurotoxicity in a reasonable time and at a reasonable cost. So the EPA has had to prioritize. And so they've done this kind of prioritization based on chemistry, the chemical composition, whether a chemical is persistent in the environment, and what its intended use is. So also another dictator is how much of the chemical is being used and where is it being used. Is it getting into your home through drinking water? And so they've reduced 80,000 to these more or less about 20,000 candidates that they need to have information on ASAP. But that's going to take some time, and we don't know that the funding's going to be there, so it's still uncertain that we'll have that information. Meanwhile, the compounds are being used. We're not exposed to any one compound at a time. And so you can think of now how complicated this gets when you have, let's say, 200 chemicals at any given time. Let's say a handful of those are chemicals that you've willingly taken. Caffeine. I see some cups of coffee. I was drinking coffee. Well, we take it with coffee and some teas and certainly carbonated beverages. But did you know that because we do that, one of the biggest environmental contaminants in San Francisco Bay is caffeine? Now, there are some people that are saying, well, what if you put caffeine, which is a neurostimulant, along with triclosan, which is coming through the wastewater, which is also a stimulant that we've identified, and you put the two together? You might have a problem. Okay? So we have to start thinking about interactions amongst these complex chemicals. And to do that, you probably can't do epidemiology. You have to take another approach. And that approach is to use models that are not intact animals, but cellular-based models. Okay. And so the way we've approached this problem, very complicated problem in the Center for Children's Environmental Health, is we said that autism's probably not just a neurodevelopmental disorder. It probably also has issues that manifest in gut, that manifest in the immune system, and that these are not independent of each other, but that they are part of the same issue, part of the same problem. And so what we have are immunologists working with epidemiologists, working with analytical chemists, and on occasion they even work with toxicologists like me, pediatricians, and what we've come up with are 3 epidemiological studies that inform the molecular and cellular approaches. The 3 epidemiological studies are called CHARGE, which is a case control study of autism 2 to 5 years of age are the recruitment criteria. Children have to be born in California, and they have to have 1 SIB. They're in 3 diagnostic groups, those with a diagnosis of autism, a developmental delay, or the typically neurotypical individuals that are drawn from the general population. Irva Hertz-Picciotto is the PI on this study. And as of today, these numbers are antiquated because they're about a year old. The current enrollment is about 1,600 families. So it's the largest study of its kind based on a case control design, epidemiological design. We gather, of course, biological samples, buccal swabs, urine, blood, but we also take very, very deep medical records as well as exposure records. Because we wanted to know how a subset of these individuals do in time as they get older, we've brought back a subset of them that were of interest to us, and that's called Charge Back. We've also, and it's not shown here, gone back in time. Now you might say, boy, that's a trick. How did we do that? Well, California is one of the few states that collects blood spots at birth. And so we have now access to all of the blood spots, at least a portion of the blood spots, for the children enrolled in Charge. So it's a heroic study. We're now in the process of trying to get it continued for another 5 years because the epidemiologists believe that they need a sample size of around 25 to 2,800 to really have the statistical power to do gene by environment interactions. But once we got this going, there were immediate concerns about what you can say at 2 to 5 if there are brominated flame retardants in the blood when the child's already been diagnosed. Well, is it just coincidental or was it really etiological? And you can't say anything if you're measuring after the diagnosis. You have to measure before the diagnosis. Unfortunately, we can't measure these chemicals in a very small amount of blood on a blood spot. So what we did is we launched MARBLES. And MARBLES is a longitudinal prospective study that when we put it in as part of the children's second project period to NIH, they were extremely, extremely excited about it, but they said, you know, we're gonna give you a good score and we're gonna give you the money, but you have a very slim chance of keeping individuals enrolled in the study. They were skeptical, as they should have been. I'm happy to say that about 4.5 to 5 years out, we have over 200 women enrolled and over 60 births. And in this design, you can collect blood, in some cases, preconception, if it's a planned pregnancy, but certainly within first, second, and third trimester from mom. And once baby's born, you can follow baby over the first 3 years, which is what we do. And at one point, we collect blood samples from baby, preferably before diagnosis, the final diagnosis. And so this gives you a trajectory in time on a subset of women that are very high risk for giving birth to an autistic child. Why? Because they already have an autistic child. Many, but not all of them, came out of the CHARGE study. Okay? So this is kind of a diagram that shows our approach. we have these epidemiological studies. The idea is that if you know what to look for or what you're asking on the questionnaire, you can do association studies, you can do logistical regressions, you can see if something contributes to risk. But of course, here you have to know what to look for, and making measurements on a large number of kids, actually finding out what chemical and how much of the chemical is very, very expensive. But nevertheless, this is essential to a multidisciplinary approach. So the other way that we've done our analysis is on cells. Cells collected from the participants in the epidemiological studies, fresh cells, frozen cells, and looked at their responses to candidate neurotoxicants in blood cells. Why? Because I want to remind you that some of the mechanisms that have been shown to be affected in autism are not brain-specific proteins. They are very widely expressed in the immune system. And so it makes sense that if you can pick up an effect in vitro on cells from kids, then you might be able to say something and instruct the epidemiologist to tune in their focus a bit. Okay, so it's a very interactive process. We also use mice cells because we can't get neurons from immune cells. It wouldn't work very well. We haven't collected fibroblasts from the kids. We didn't even think about it because the method didn't even exist when we started this study. The idea there would be if you could collect fibroblasts, you can then reprogram the fibroblast to become neural cells, and then you could study the neural impairments in a dish from children with various forms of autism. And unfortunately, we don't have fibroblasts on these kids. In marbles, we do have something even better. We have stem cells, and that's the way we're going to go forward with that. Okay. But so we use neural cells derived from mice or from rats, which serve as a surrogate. And some of these mice and rats are what we call wild-type strains, and some of them have the same mutations that have been implicated in autism. Okay? So we put all of these things together, and what we're hoping to do is identify this risk assessment ratio. So in order to have something change at the level of governmental banning or limits to exposure, what you have to do is you have to come up with a risk assessment. And unfortunately for autism and other neurodevelopmental disorders, the only way you assign susceptibility is once you find the no effect level for the general population, the risk assessors just divide that level by 10. That's the best they can do. So they say, if you divide by 10, then you're including minimal risk for susceptible populations, such as kids or individuals that have a genetic propensity for harm. Clearly, everybody realizes that this division by 10 is very, probably it doesn't protect everybody. Okay, so. Is something going on? This is work from Hertz-Picciotto last year, the year before last actually, which looks at birth cohorts between 1990 and 2003 as birth cohorts, and how the rates of cumulative incidence per 10,000 increase between the ages of 1 to 9. And it's very clear. This is based on the California database, the DDS records. And what you see is that in 1990, compared to 2002, the rates, especially for younger children developing autism, has increased dramatically. So that's a fact. Now, what's driving that? That's a big question. So some of the analysis that you can find in this paper suggests that it can't all be accounted for by what we call diagnostic drift, diagnostic substitution, or the fact that maybe instruments have changed over time and become more inclusive. Another possibility, of course, is that you know that if a doctor gives a patient an autism diagnosis, it opens up certain level of support from insurance companies as well as government. So, but regardless, this is not all due to something that we know of. Possibly environmental changes are contributing to these increases in cumulative incidence. So I want to give you results from a study. Mercury, big issue. We weren't and we haven't set out to answer the question, does mercury cause autism? That's a very complicated question, okay? So the data I'm going to show you asks a very different question. And it does have ramifications, immediate ramifications. A very large number of parents, once they receive an autism diagnosis, or if they already have an autistic child and are having another one, do blood tests for heavy metals, as well as other things. And the results you get back from those analyses often dictates what you do about it. So if they come back in the 10th percentile and it's below the national average, typically you don't worry about it. Or maybe you do. Some people still worry about that. But if it comes back in the 90th percentile, where your levels are higher than 90% of the general population, you start to worry. And what's one thing that you can do to respond to that concern? Well, you think about chelation, okay? And so, unfortunately, we don't know what the outcomes of chelation are. There's never been a double-blind placebo test on chelation therapy. And so, we don't know whether it does nothing, whether it causes harm, or whether it has benefit. We really don't know. But chelation is a big issue. So, at some point back, I think about 5 years ago, the National Institute of Mental Health launched a chelation study. And that took a while to get past the institutional review boards, several of them. We were going to measure the metal levels for them. And so we measured metal levels in the vitamins that they were going to supplement the participants, and they were clean. The Flintstone vitamins that have all these different colors, you know, because you wonder if there's traces amounts of metals that get in there that shouldn't be in there. They were fine. And we also got the chelating agent that they were going to use. And there we actually found some very low levels of mercury. Nothing that would do harm, but it's certainly, if you didn't account for it, would drive you bananas when you're measuring urine or blood levels, right? Because you're actually putting in mercury with the treatment. And so you had to account for that. And that wasn't harmful levels. I want to make sure you know that. But these chelates are very thirsty for heavy metals. And so even if there are traces amounts in the air, they grab onto it. And so you had to, but it didn't matter because a study came out of Cornell that said chelation in the absence of heavy metal intoxication causes harm in rats. It immediately stopped the study. So we still don't know. So what we did was we went out to say, if we take a very well-documented case-control design, do children with autism at a point in time have higher, on average, higher blood mercury levels than their counterparts that are in the typically developing group? It's a very simple question that needed to be answered. So what we did was we took a total of 566 participants in 3 diagnostic groups: autism, no autism with developmental delays, and population-based, typically developing controls. The mothers were interviewed about household, medical, and dietary exposures. Major exposures to mercury come through fish, amalgams, and to some extent vaccines. But by the time we got this study off the ground, vaccines were already being weaned from childhood vaccines. So there was only a fraction of the kids that had some of the vaccinations that contain thimerosal. The blood mercury was measured by the best methods available to date, which is an inductively coupled plasma resonance mass spectrometry, very sensitive, very wide dynamic range with lots of standards and blanks. And then the epidemiologists put this data together and basically did logistical regressions or multiple linear regressions to say, first of all, are there differences in mercury levels amongst the 3 diagnostic group, and what's causing the difference in levels? And if you correct for it, are there real differences? So what happens is that, as you might expect, there are some kids with very, very low levels of mercury, some kids with relatively high levels of mercury. There were a couple of times when we actually had to send letters to say you need to get a second test done and act appropriately if, in fact, those were over the limit. But if you look amongst the 3 colors, in general, there was really no striking difference in blood mercury levels in these 3 diagnostic groups. But what is interesting, and please keep in mind, that the ranges of exposure are quite broad. So if you were to look at, for example, calcium or magnesium, nutritive metals in these kids, the variation would be very, very tight. So these are clearly environmental exposures. What dictates the environmental exposure? What's the major environmental exposure relative to where these kids sit on the spectrum of mercury? Basically, the total blood mercury was neither elevated or reduced in charge relative to NHANES, which is general population study across the country, that the DD group had lower levels even after correction for exposures, but the AU group did not. And fish consumption and dental amalgams, as expected, would be the best predictors of where the kids were on that spectrum of mercury levels in the blood. So basically, this says nothing about whether mercury at an earlier age promoted dysfunction or contributed to autism. It says that at a time when a lot of kids are being chelated, is there really a reason to chelate them? Okay. And this study would suggest probably there isn't a compelling reason to chelate these kids because their mercury levels are way below the 2 microgram per deciliter standard that's going to be the new standard. Right now it's 10, okay? Other people in the center were asking a very different question. They said, wow, we've got this broad distribution of mercury levels in this population, and we have their controls, and there doesn't seem to be a difference in their distribution or their means, but let's use high throughput transcriptomics to say, if you look at the mRNA in these kids, and we know that there are differences between AU and TD, is the difference correlated with the mercury level that's in the blood? So this is a very different question. It asks, sure, okay, there is a wide distribution of mercury levels in kids that have autism, but they're virtually identical to those that don't have autism. Do the autistic kids respond differently to their blood mercury levels than do the typically developing? That's a more toxicological question. It's not cause and effect, but as if since the mercury's there, how do the 2 populations respond differently? And there we found some interesting observations. So, this is the experimental approach, and again, we measured the mercury levels, of course, and then what you did here was you took extracts of RNA from the same kids and put them on a gene array, a transcriptional array, which essentially queries the expression levels of about 20 to 30,000 genes. Some of those, it's probably closer to 20,000 genes that we know functions for. And there, you can ask the question, you can build up these models, how many of these genes change with mercury significantly in the general population, but not in the autistic population? And of course, you can ask the same question about the autistic population, genes that change with mercury concentration in the autistic population that don't change in the typically developing population. And then you can do an interaction term. To make a long story short, the real interesting parts were that there were genes that changed in the GP that didn't change in the typically developing general population, that, and genes that changed in the autistic group that were not concordant. In other words, they were different genes, okay? But what's really interesting is if you look at the intersex here, this is where we can make some observations. There were 26 genes, and that's a small number actually, that correlated with mercury levels in both the autistic population and the typically developing population. And this was limited to boys for this study, this little pilot study. 11 of these genes, 11 of the 26, were different between the two groups. In other words, if a gene went up as a function of mercury in the autistic kids, they might have gone down with the typically developing kids. And so they changed in different directions. And so we asked, what are those genes? What are the functions for those genes? And it turns out that many of these genes were correlated with important things like cell death and cell morphology. Well, cell morphology is one of the things that has interested biologists, neurobiologists in autism, because it involves synapse formation, and some of these genes are involved in synapse formation. There were also a subset of genes that were involved in amino acid metabolism, cell morphology again, but also antigen presentation. And that was important because we had studied the effects of mercury on a major antigen-presenting cell called the dendritic cells in mice. This was the conclusions of this study. But we had studied dendritic cells in mice and shown them to be amazingly sensitive to organic mercury. They are very much altered in their ability to respond to oxidative stress as a result of very short mercury exposures. And we think this is important mechanistically because it's not saying that, you know, when you have mercury, everything goes bad. It asks, when you put mercury in a particular location and cells that hone in on, let's say, the antigen that's presented with that mercury, what cell would be most susceptible? And in this case, these dendritic cells are in fact the key homing cells that go after whatever's been injected or whatever you've been exposed to through other means. So, of course, we proceeded to test this in real mice. And this is where I started to get skeptical about mice, not because this was a negative result. I think this result was very important. But the idea that we chose a mouse that had a propensity for immunological problems, especially adverse response to mercury. So we call this a susceptible strain. Why? Because one can break tolerance very easily in this strain and promote autoimmunity, which we think is involved in autism. But the way this mouse is autoimmune prone has nothing to do with autism. In fact, we don't quite understand how this mouse gets autoimmune problems in response to mercury. We have some ideas, but the genetics is not well understood, and the genetics don't map up to any autistic population that we know of, okay? But what we found was when we first started testing these animals, their behavior was already different from the typical mice. They're much more aggressive. They don't seem to have a problem with being in an open T-maze, you know, a measure of anxiety. So their behavior is actually not your standard behavior for a mouse. And so you sort of wonder that, well, then you stress them out with injections and antigens and mercury, and do you see anything? Well, the bottom line is that we saw nothing. Okay, so we exposed these mice to a neonatal exposure to vaccine antigen and mercury, subdermal, as neonates, but there were no behavioral outcomes that we could really hang our hats on. So that kind of was a dead end. Okay. But that's not to say that children with autism do not have immune presentation. Their immune systems are very, very different from typically developing, and the data on this is going astronomical now. If you type in autism and immunity, you'll find, in PubMed, you'll find many, many hits now. And these are more recent papers than older papers. So the interest in this is really quite is going up year by year. But at the Mind Institute, Paul Ashwood and Judy Vanderwater have actually made some compelling discoveries about the immune systems of autistic kids. And I'm just going to show you a couple of them here. First of all, that there is a problem in a particular kind of immune cell called the NK cell, which is very much involved in marshaling immunity. And there seems to be a hyporesponsiveness of these NK cells. So if you sample the blood from a child and you say, what are the markers like? Well, they're elevated. So there seems to be more NK markers in the serum of these kids. If you collect the cells and put them in a plate and challenge them in vitro to antigens, they actually respond very poorly. So it's almost like they're hyporesponsive, and there's some sort of reciprocal mechanism to try to increase their numbers. So we're still trying to figure out what that means, okay? In addition to immune issues in the children, there also seems to be a subset of women with maternally derived autoantibodies that persist long after they've given birth to an autistic child. And these antibodies are to brain-specific proteins. Well, since IgGs cross the placental barrier during gestation, it's possible that these antibodies are having an effect on the developing fetus. Now, how these antibodies arise, why they arise, that's very hotly being pursued right now. We've identified 3 of the antibodies and what they bind to, and it's quite interesting. And so that I can't say very much about now because it's premature, but stay tuned, okay? So this also led us to a better model for immunology and immune model of autism, and that is to take these antibodies in the form of purified IgG and expose mice during gestation, just like an autistic child would during gestation, or one that eventually is diagnosed with autism, and say, now do we see behavioral outcomes? This is a very important model because if in fact the IgG alone produces behavioral deficits in the offspring, that could give you a real clue of what's going on in the subset of women that have these autoantibodies. Yeah? No. These are absolutely what we call wild-type mice. Yeah, except that they're inbred and humans are not. But we had to start somewhere. But they don't have any propensity for autoimmunity. So this is very important for several reasons because autoimmunity involves a huge amount of oxidative stress. We know that challenging young children with vaccines is essential to prevent communicable diseases, but there may be a subset of kids who can't process the vaccinations, especially when multiple vaccinations are given at the same time. So you might say, well, that's just hand-waving. But in fact, we know of genetic mutations in, for example, calcium transporters, which in fact are very, very non-clinical until you either get a flu infection and there's a high fever, or in fact you have an adverse response to a vaccination, which causes these recurrent seizures to occur, and this could have a drastic effect on the development of a young child. So we have those examples, but that's not to say that this is driving the major prevalence of autism. But it is a hypothesis that can be tested. And one of the reasons that we're interested in this hypothesis is this idea that children with autism are under chronic oxidative stress. I was very skeptical when that initial sort of view came out and a little bit of data came out, mainly because if you read the literature, virtually every disorder is associated with chronic oxidative stress, whether it's schizophrenia or Parkinson's disease, or you can go on, and non-neurological disorder are associated with chronic oxidative stress. So what is chronic oxidative stress? Well, that means that you generate reactive oxygen species and make heavier demands on your antioxidant system. And where this all originates is in the mitochondria, because the mitochondria is the single most prolific generator of reactive oxygen species, especially when something goes wrong in oxidative metabolism. And so we did take a look at actually a small number of kids in a pilot study that were enrolled in Charge Back, and we called them in for specific reasons so we could keep the number down. These cells from the kids had to be rushed to the lab within 4 hours from the site of collection, and so that was a challenge. But in fact, what we found was not only there was evidence for mitochondrial impairments in kids with autism relative to case control typically developing, but that in fact the cells in the blood generated more reactive oxygen species. And there were very interesting effects. So we did a very large number, actually, that didn't end up in this paper, looking at their mitochondrial DNA copy number. This was Cecilia Jalevi, who's a mitochondrial expert. We convinced her to be interested in this project. She did. And she has probes that actually measure the mitochondrial DNA load in these blood cells. And these cells, these mitochondria change their copy number as a physiological response to energy needs. So when you have a change in energy need, you need more energy, these mitochondria will replicate their DNA and you get increased oxidative capacity. That's a normal response. But what we found was that in fact, there was increased oxidative stress because there was higher reactive oxygen species in these cells. And in fact, we saw much higher mitochondrial DNA numbers per mitochondria, but in fact, oxidative phosphorylation was not concomitantly increased. So in other words, this response is a pathophysiological response as opposed to what you would consider a normative response. And so we're trying to track down what aspects of mitochondrial dysfunction relate to autism. It's probably not any one, there are probably several. And other people have now started looking at this big time. So why do we need to identify environmental risk factor for autism? You can think of many genetic disorders that we know exactly the one gene that causes the genetic disorder. Let's take an example, muscular dystrophy, Duchenne's muscular dystrophy. There's one protein that's missing, it has a premature stop, the kids are very adversely affected. We know what the gene is. We've been trying to put the gene back for over 25 years now. Hopefully there'll be some progress in that soon, but it hasn't panned out. In autism, because of the latest data, we think that there are environmental triggers or environmental modifiers. We need to identify them. If we can identify them, even at the level of family susceptibility, we can limit exposure, and that should help. In fact, if you look at the highest quality risk factors for autism, genetic risk factors, or cell biomarkers for autism, and ask, what's a common convergence? In fact, one can identify convergent factors. You just have to look at it from a mechanistic perspective, and then hopefully that information will be used by population-based studies to really dig into what's important and maybe ignore what's less important. Because many of the mutations, and in fact many of the environmental chemicals, can affect common systems, signaling systems, in both the immune system, the peripheral immune system, the central immune system, which involve the microglia, but also the neurons. And I'd like to give you just one example of that. So this is the kind of, so in the nervous system, now we're back up in the brain, the leading hypothesis for many genetically derived disorders that have very high autism rates is that in fact they all result in an imbalance between excitation and inhibition of the nervous system. So that could be mediated by factors that are immunological, cytokines, in the brain, but they could also be due to misprogramming or misfiring of the neurons in the central nervous system. If you have an imbalance between excitatory, green, and inhibitory, red, neurons in the brain, or they don't form proper connections, this could lead to several aspects of autism, including a deficit in social reciprocity, but clearly things like epilepsy and what we call an immune response in the central nervous system, or neuroinflammation, okay, which could contribute to impairment. So what chemicals? Well, when these data started coming out, and they're now not that new, toxicologists would have told you it's a no-brainer. We know chemicals that affect these systems that are clearly contributing to autism risk. So GABA receptors are in fact the major inhibitory neurotransmitter postnatally, and during development they actually are excitatory. But clearly they play a role in proper development of the nervous system because they control excitability. We know that if you look at the level of epigenetics, complex gene-gene interactions, or even polymorphisms, that there are pieces of evidence that say those systems are impaired in autism. Most recently, Chow et al., this was one of the papers that made finding of the year for Autism Speaks last year, that in fact dysfunctional GABA signaling mediates autism-like effects in the Rett syndrome, both in humans as well as in mouse models. And they did a very, very, very detailed study here. Okay. So why does this make sense to a toxicologist? Because there are chemicals that we have put out in the millions of pounds that are in fact neurotoxic to insects because they're GABA receptor blockers. So it's a common mechanism. If you have chemicals that get to the central nervous system and further impair GABA signaling when there's already an impairment, one would think that those children would be much more susceptible to the seizure-genic effects of these pesticides or these insecticides. And so Roberts from the Department of the DDS, I think, anyways, from one of the state agencies in California, actually tested this hypothesis using existing databases. And what this shows is that, in fact, one set of chemicals seemed to increase risk for two women that were near agricultural fields that were sprayed with a particular kind of insecticide. They're known as dicafol and endosulfan. These have been known for years to work through GABA receptor blockade. And what was really compelling about this study was that as the women were farther away from the agricultural fields where these compounds were used, the risk factor declined. So there was a dose-response relationship. Now, dicafol and endosulfan are of historical interest. They're still used in California. Their big brothers, endrin, dieldrin, ketone, not paraquat, but ketone, all of those have been banned anywhere between 75 and 1988 or thereabouts. Problem is that those compounds are extremely persistent, very persistent. So if we measured levels in our blood right now, even though they haven't been used for a while, you'd pick them up. Endosulfan and dicafol are not the major insecticides in agricultural use, but the fact that they picked up this increased risk is quite notable. But from a mechanistic perspective, you say, well, you know, I'm not surprised. So we got away from these kinds of very persistent insecticides, and we now have newer, safer insecticides. So let me point out one. Do you all know what this one is? It's 4-alkyl-1-phenylpyrazole. Yeah, I know, it's got a very simple name. And it was biorationally developed because it only blocks insect GABA receptors. Well, things have gotten more complicated because the more research you do, you know that in the mammalian nervous system, there are 15 gene products that make GABA receptors, and we're not quite sure which get turned on and which get turned off exactly during development. So there's one in particular called the beta-3, and that should strike a chord, because if you go back in your notes, that beta-3 is a very important one because it's the one that promotes most of the negative inhibition that keeps you from going into seizures. Mutations in this one does cause seizures and, in fact, can cause sleep disorders. And so about 3.5 years ago, I had to change what I say about this to the students I teach, because I used to say, wow, this is really fantastic. You can really have great control on your dog for 3 months and have no problems because it doesn't affect the mammalian nervous system. And just as about I was getting comfortable saying that, a lab in Berkeley basically showed that it cannot discriminate between that beta-3 type GABA receptor, which is a major one in developing and adult humans, and the insect receptor. Basically, it latches onto both just as well. And that's new knowledge. And so the question is this particularly potent on children that already have a deficit in GABA receptor signaling? And that's a hypothesis that is being tested now. More common are the pyrethroids. And we heard about it at the beginning of the talk, that pyrethroids are thought to be more safe than all of the previous insecticides because they break down more readily in the environment. But it can't say very much. The initial abstract came out about 2 years ago at IMFAR in London. And before Dr. Picciotto landed in London, the London Times was calling anybody they could get a hold of to say, pyrethroids cause autism? And all I can say at this point is that our results suggest that, in fact, these are a risk factor, especially when you look at a particular gene variant in a metabolic enzyme that may be involved in getting rid of these guys. Okay? So that's all I can say right now because it hasn't undergone peer review. And then finally, what's calcium have to do with autism? Well, we all know about calcium is good for you. You need to have calcium intake for strong bones. But what we're talking here is not calcium outside the cell or in bone tissue matrix, but in fact calcium that's inside the cell that serves a signaling function. When we evolved out of the primordial ooze, calcium was very toxic to the cells. And they did everything to evolve proteins to push it out of the cell, and then they got smart and they actually used it to do everything from regulating signal transduction, the level of metabolism, gene transcription, growth, mitigation, migration, and cell death. In fact, we call calcium the common currency in cell signaling. All cells use calcium signals to go about their normal business. What was really interesting to us is that if you start to make a list of those risk genes that contribute to autism, they're all either calcium channels, transporters, or regulated by calcium. Now, you might say there are over 2,000 or 5,000 proteins that are regulated by calcium, but I guess we're driving home the point there. That if you have a common dysfunction by environmental chemicals, individuals with these genetic dysfunctions would be more susceptible. And so let's pick a gene that's been highly associated with autism, the MET gene. Pat Levitt and his colleagues have clearly shown that MET gene polymorphisms are associated with autism risk. Well, the MET gene, in fact, is the receptor for hepatocyte growth factor. We think that because it's called hepatocyte growth factor, it only works on the liver, right? Not true. In the central nervous system, that signaling cascade is very important for dendritic growth and forming networks, especially in the hippocampus. And this is just an example. Here's a cell that's received vehicle early and late. Here's a cell that's received hepatocyte growth factor early and late, and you don't need math to say it's causing a hypertrophic effect. And this signaling cascade would be expected to be highly regulated because it's a physiological response, right? The way this happens is that, in fact, the signaling system initiates a rise in intracellular calcium, which promotes this growth. So calcium is pivotal. It's the pivotal next step to receptor activation. Timothy syndrome, okay? Long QT. Colleagues at Stanford just published this paper where they took fibroblasts from autistic kids that have Timothy syndrome, the mutation. And remember I said that the clear presentation is long QT, arrhythmias. They reprogram those fibroblasts to become heart cells. The heart cells beat. And if you compare cells from a typically developing individuals and one that have Timothy syndrome, you can clearly see that there's irregular firing. So they've maintained the penetrance of that gene even after taking it from a fibroblast to a muscle cell. And you know where they're going to go next, right? They're going to make neurons out of these inducible pluripotent cells. And that's going to be very, very telling. So, calcium is important. These compounds, very highly regulated. They include dioxin, which you've all heard of. Anything that has dioxin-like activity has very hard time making it to market. So, what kinds of compounds are dioxin-like? Certain PCBs are dioxin-like. Okay? Polyaromatic hydrocarbons, which affect the MET gene, as does dioxin, are dioxin-like. Well, it turns out that dioxin and polyaromatic hydrocarbons and these PCBs can regulate the level of transcription of the MET gene. We're starting to piece gene-environment interactions together. What about these compounds? These are not regulated. These are the ones we work on. We'll let somebody else work on all these, but these are already regulated. Somebody has this kind of activity or this kind of structure, very hard to get them into the home, much less into production. These look different, don't they? They don't have that flat appearance. They're called non-coplanar compounds. Some of the PCBs found in the environment are non-coplanar. So when these things were first put in the environment in ballasts for lighting fixtures and making oils more heat absorbent, both of these were in the oil. They were just really messy mixtures of compounds. These degrade over time. Takes a long time, but these tend to disappear. These are the ones that make up the majority of what you find just out here in San Francisco Bay sediments. But nobody's worried about these. Well, it turns out that these are the ones that target abnormal calcium regulation in cells. And they're very potent at doing it. But in fact, because nobody cares about that basic non-coplanar structure because it doesn't have dioxin-like activity, a lot of things have gotten under the fence. So what are some non-coplanar structures? Triclosan. Flame retardants, triclosan, bisphenol A. These are all non-coplanar structures. This is the PCB here, that have very little to no dioxin-like activity. Are these a problem? We haven't been regulating them. Obviously, the flame retardants we're just starting to regulate. Triclosan, I think the companies have made a very, very good case for why they're absolutely necessary to put in your underwear and your pizza cutters and your cutting boards, in your Tupperware containers, in your hand soap, in lipstick, and I can go on and on. They're everywhere. You can buy a ton of triclosan over the market on the internet for less than 10,000 bucks. Now, you can't sell it and do something with it. You need a license to do that. But very, 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 very widely used compound. We found the activity of this based on our work with this structure. Scoured the literature for data on the ability of this compound to be toxic. And in fact, if you look at all of the literature, for it to be acutely toxic, you basically have to either feed the animal a huge amount, way beyond what would be considered reasonable, and in terms of dermal exposure, It's not a problem. If you were to breathe this stuff in, however, there's some indications in the literature that it may not be such a good thing. We tested this in a living mouse model, but we didn't want to expose them orally and we didn't want to expose them dermally, which is the right way to expose them because that's how we're exposed. And believe me, there's now CDC data that says this is in all of our bloodstream, so it's not like we're not exposed. We did an intraperitoneal injection at what we thought was 1/10 the problem dose that you see orally. So instead of 5 to 6 grams per kilogram, we administered 100 milligrams per kilogram IP. The rat was being monitored for hemodynamics. And this is not published yet, and the company has come to see me. The mouse died within 10 minutes of heart failure. We would have predicted that based on the mechanism that we had identified in cells. Now you ask the question, why is it that when you wash your hands, your heart doesn't stop, or at least get weaker? We think we know the answer. This chemical has hydroxy groups on it. When you're exposed orally or dermally, and this is where your defenses really become important here, orally or dermally, the transfer rate into the bloodstream is relatively slow. It's continuous because you're being exposed all the time if you use products with the compound in it. But it's relatively slow. As it enters the bloodstream, it's one of the most rapidly glucuronidated structures that we have seen. It just, what's called a phase 2 metabolic reaction. You get glucuronidase added onto the structure, and that makes it inactive. And so the blood levels are there, but they're in an inactive form. And so one needs to ask, what if you were impaired in glucuronidation? And we know several polymorphisms where that is in fact a fact. Or what if you're really young and you haven't developed expression of your glucuronidase transferases, the enzymes that do the reaction? So what we're suggesting here is that we really need to study the way in which these compounds work. And now we need to take it back and ask, how do they affect the balance of excitatory and inhibitory neurotransmission in the nervous system? And now epidemiologists can maybe focus a little more on this compound and maybe a little less on mercury. Thank you.
The UC Davis Mind Institute was created in 1998 with a promise to find cures for neurodevelopmental disorders. Every day, our physicians and researchers come closer to fulfilling that promise. Their groundbreaking research on autism, Fragile X syndrome, chromosome 22q11.2 deletion syndrome, ADHD, and other brain disorders are helping children achieve their fullest potential. Please visit our website to find out more about current studies, upcoming events, and how you can help make a difference.